Youth Biolab Jeunesse.
Hi, I'm Steve.
I'm Megan.
And I'm Angelica.
We're from the Youth Biolab at the Albrechtsen Research Center at St. Boniface Hospital. We all know the job of the hospital is to treat patients. Here at the Research Center, our job is to study disease. If we ever want to cure a disease, we have to understand it first.
When you think of scientists studying disease, you probably think first of things like cancer or more recently, COVID-19. We don't study either of those here. But there are research scientists over at the National Microbiology Lab who are studying infectious diseases like the flu or COVID-19 or Ebola, where they have special equipment that can protect them from the germs that they're working with. There are also groups of cancer scientists working over at Cancer Care Manitoba in Health Sciences Centre, and it's their job to study cancer. We like to keep scientists together so that they can work as a team to come up with cures or treatments for these diseases that we see.
At the Albrechtsen Research Center, we have three big interests. On our fourth floor, we have the Division of Neurodegenerative Disorders. This is where we study diseases that affect the brain and nervous system, just like Alzheimer's disease and type 2 diabetes nerve damage. On our third floor, we have the Institute of Cardiovascular Sciences. This is where we study diseases that affect the heart and cardiovascular system. On our second floor, we have the Canadian Centre for Agri-Food Research in Health and Medicine, and this is where we study food. They research foods that are really good for us, some that might be bad for us, and some that might be so good that they act like medicine.
Our lab, the Youth Biolab, was created so that we can share some of the things that we do here in the Research Center with you. And hopefully we can help you learn a little bit more about how medical science works and understand a little bit more of the science of your own health. We're here on the fourth floor of the Research Center, where we have the Division of Neurodegenerative Disorders. When I say neuro, we're talking about neurons or brain cells, and degenerative means we're talking about diseases that break down those nerve cells. These nerve cells could be in the brain, or they could be elsewhere in your body, like sensory neurons that you use to touch and feel. Here at the DND, we're really interested in major neurodegenerative diseases like Alzheimer's disease and diabetes, and our labs study many aspects of brain health, dementia, nutrition, memory, and aging. These are obviously very complicated problems, so today we're going to focus on Alzheimer's disease and how our scientists study it. You've probably heard of Alzheimer's. It's the most common type of a range of memory loss problems that we call dementia, and right now there's no cure for it. Alzheimer's disease affects memory, thinking, and behavior, and it often starts with people having trouble remembering things that they've recently learned, and it can get worse and worse to the point where people can't recognize family members and get confused about events and places. Eventually, people can forget how to breathe and swallow. Our scientists here are trying to figure out what's actually changing in the brain to cause all of these memory problems. If we can understand what is happening, then maybe we can figure out a way to fix the problem. As you can imagine, we don't really understand how memory works in the first place, so studying a disease of the memory is very complicated. However, scientists around the world have been piecing this complicated puzzle together over many years, and we're starting to see what that puzzle looks like. Let's head down to the lab and show you how we study these things. When our scientists want to study a disease like Alzheimer's, there are many ways we need to go about it. One of the big problems with Alzheimer's is that we can't really detect it until a person starts showing signs of memory loss. And by that point, it's really too late to reverse the damage. So we need to understand what's actually happening inside the brain if we want to figure out how to prevent or treat the disease. Brains aren't easy to study. They don't have moving parts like a heart or an obvious way of looking at them to see how they work. A lot of our understanding of how the brain works has come from examining brains of people who have had different diseases in their brain and comparing their brains to healthy brains. We're getting better at understanding the brain using techniques like MRI scanning, but to truly understand a brain, we have to understand how they work at the level of the brain cell. So your brain is made up of billions of brain cells, or what we call neurons. And although there are several types of neurons, their basic function is to send information from one place to another. When we compare an Alzheimer's brain to a healthy brain, we start to notice that there are changes in how those brain cells are doing those important messaging jobs. This doesn't happen everywhere in the brain, but it seems to start in specific areas that are very important to memory. So when we compare those Alzheimer's brains to healthy brains under the microscope, we start to see changes in one special area. It's not in the front of your brain, the back, or the sides of your brain, but a little part of the brain buried deep down inside called your hippocampus. You have two hippocampi, one on each side of your brain. In you and me, it's probably about the size of your baby finger, and they're buried in behind your ears. The hippocampus is one of the most important parts of the brain when it comes to memory. It's involved in forming short and long-term memories, and it helps you understand your location in space. You don't store a whole bunch of memories there, but it's really important for forming those memories and deciding if you should keep them. If you think about parking a car in the parking lot at the mall, it's your hippocampus that helps you remember where it is when you come out. Do you need to remember where you parked that day for the rest of your life? Of course not. That's your hippocampus at work. It doesn't make a long-term memory out of that. But if you came out of the mall and your car was on fire, would you remember that for the rest of your life? Probably. And that's your hippocampus at work too. It'll store that somewhere as a long-term memory and maybe even wrap in emotions like fear or sadness with that memory. Every memory you have, whether it's a memory from a long time ago or just from the start of this video, has gone through your hippocampus. We don't really understand why we see these changes in the hippocampus in Alzheimer's. It might be because of diet, brain injury, or connections to other diseases, and it seems there might be some genetic cause for it. Whatever the cause, we know that when we look at the cells of the hippocampus in the Alzheimer's brain through our microscopes, they're different from cells in a healthy hippocampus. So let's talk about brain cells for a minute so you can see what we're talking about. You might have seen a picture of a typical cell that looks something like this. Brain cells don't really look like this type of cell, although they might have many of the same parts, like mitochondria, nuclei, membranes, and things like that. Brain cells are structured so that they can send messages over long distances, and to do that, they need to have long branches that can reach other cells. So here's a model of a brain cell that we like to use when we talk about how brains function and how brain cells do their job. Like we said, brain cells aren't your typical round cell. They do have what we call a cell body, which is where all the nucleus and a bunch of organelles would be. But they have this whole network of branches that help them receive and send messages. You'll notice there's some branches coming off the cell body that we've got here in yellow string. These are what we call the dendrites. Typically, this is where the brain cells will receive a message. You'll also notice a big long tail coming out of the cell body, which we call the axon. And this is how a brain cell is going to transmit a message to another cell somewhere. One brain cell can't really do anything all by itself. It needs to communicate with other cells. That could be a muscle cell in your fingers as you send a message to move your hand. Or it could be another brain cell somewhere else in your brain if you're encoding a memory or running some other sort of brain function. So let's bring out another brain cell and we'll show you how this works. So now we've got another nerve cell. And our job here is we want to try and send a message all the way from here, all the way down to the end of the table using nerve cell functions. So when a nerve cell is going to try and send a message, there's a few different things that need to happen. The two main ideas in sending a message in a neuron is we need an electrical signal and we need a chemical signal. So first, if this cell is going to send that message, it will start an electrical zap that runs down through the axon. In this case, we're going to represent that electrical zap with this roll of tape. So you can imagine this nerve cell sending this tape, this electrical zap, all the way down to what we call the synaptic terminal here. This is the same thing that happens when you send a message down to your toe to tell your toes to wiggle. You're sending an electrical message that'll then create a chemical release. So in this synaptic terminal, it's loaded with little test tubes. And these test tubes are here to help us represent the chemical release. These are what we call neurotransmitters, and they're special chemicals that signal to brain cells to do something. You might have heard of some of these. You might have heard of dopamine or serotonin. These are some of your mood chemicals. Dopamine is a happy chemical. So when you're feeling good and excited and happy, there's a lot of dopamine being released in your brain and stimulating other nerve cells to create that mood or create that feeling. You can have imbalances in these things. We've heard of depression and things. And those are changes in those chemical levels. And we can use medicines to try and get some of those chemicals back in balance. So let's bring out Angelica and we'll show you how this all works in action. So now we've got Angelica and she's going to help us demonstrate how we send this message between one nerve cell and the next. Remember, there's two things we need. We need an electrical signal, we need a chemical signal. Once that chemical gets released to our next nerve cell, then it will get excited enough to send that message onwards. So Angelica is going to pick up our synaptic terminal there, and I'm going to get ready to send that message. So as soon as we fire our electrical signal, we get a chemical release that will then stimulate the next nerve cell to send along the signal. One other important part of nerve cell function is what we call reuptake, and that's what Angelica is doing right now. She's taking those chemicals and putting them back into that synaptic terminal so they don't keep on stimulating that nerve cell over and over and over again. So that was nice, normal, healthy nerve cell function. You have your electrical signal or your action potential, which creates a chemical release or a neurotransmitter release that'll then go on to stimulate another nerve cell or another cell somewhere in your body. When scientists start looking at Alzheimer's brains, we see changes in the way these things work. So let's have a look at our nerve cell here, and I'll show you what scientists notice when we start to examine Alzheimer's brain cell function. One thing that seems to pop up in those nerve cells of the hippocampus are things that we call tangles. It's not necessarily a tangled axon, but it's kind of what it looks like under the microscope. It's more damage than an actual twist or tangle, but here we can represent it by putting some knots into the axon. The other things we see in Alzheimer's brains, in that Alzheimer's hippocampus, is what we call plaques. And what plaques are big blobs of protein that form between the end of one nerve cell and the start of another in this synaptic junction here. So here's a nice big blob of protein. And you can imagine if there was a big blob of protein here between these two cells, it's going to cause some problems in that nice normal nerve cell function. So now, if we're going to try and propagate that signal again, if we're going to try and make that release to our next cell, you can see we're going to have some trouble. As this comes down, the axon is damaged and tangled, where we aren't going to be able to make that signal go. Even if that signal was able to get through, these blobs of protein here, the plaques, are going to stop those neurotransmitting chemicals from getting to our next cell. And whatever that is, that could be memory encoding, that could be making a new memory that we rely on our hippocampus to do, it's just not going to happen. And once these cells are damaged to this point, they'll eventually die. And once they die, they won't come back. Scientists aren't really sure why those plaques and tangles form around those nerve cells in the Alzheimer's brain. So we spent a ton of time trying to figure out how, where, and when these things appear in the disease. One of the things we do know is that they tend to appear first in the hippocampus. So it'll help us if we can actually look at a real brain to see where that hippocampus is and what it looks like. The brain that we're going to have a look at today is a sheep's brain. We don't study sheep here at the research center, but their brain's a really good model of the human brain. It has a lot of the same parts. Normally we say our brains, are about the size of two fists. So the sheep's brain is going to be a little smaller and a little different in some areas, but still a sheep has to remember things. So it has a lot of the same memory functions as we do, and it has the two hippocampi just like we do as well. This sheep brain has been preserved in a chemical that's going to hold it all together. If we were to take a brain right away fresh out of an animal, it would be soft and mushy and kind of greasy. So we put it in a chemical that will hold everything together for us. A few rules as we do any sort of dissection or when we work with anything that comes from a living thing, whether it's live, dead, preserved. The main rule here is we need to treat these things with respect at all times. Whether this is cells from an animal, whether this is an organ from an animal, throughout any of these things we need to treat them with respect. That means when I go through this, I'm going to go carefully. I'm going to take care with what I'm doing. And our main rule here is we never say the word gross, we only say interesting. So let's get our brain and have a look and see if we can find that hippocampus. So here's our sheep's brain. If you look at it, you'll notice it has some of the similar parts that you might have seen in a brain diagram before. We can kind of figure out where the top, bottom, front, back, and left are. We've got the front of the brain here. The back of the brain we can tell. We have this lump here called the cerebellum, and your spinal cord coming out the back. In you and me, obviously, the spinal cord comes down because we're walking around on two legs, whereas the sheep's spinal cord would come out the back because it's walking around on four legs. One thing you'll notice as we look at the top of the brain here is it looks like it has almost a plastic coating on top. This plastic coating isn't plastic. It's actually something called your dura mater. Your dura mater is one of three layers of what we call the meninges, and these are a layer of membrane that coats the brain. The dura mater will protect it from rubbing against your skull. The other layers will help hold in the cerebrospinal fluid that wraps around the brain to cushion it inside your head. Let's see what else we can see if we turn towards the bottom of the brain. So on the bottom of the brain, we see our spinal cord, but also the parts of the brainstem, your pons and your medulla oblongata. These parts of the brain are very important in regulating kind of all those basic functions that have to happen all the time, like breathing and heart rate and temperature. A lot of the very basic things that you don't have to think about controlling in your daily life. We can also see a couple other things. This little lump right in the middle is the pituitary gland, and it creates hormones that are responsible for controlling growth. One more thing we can see is what's called the optic chiasma here that leads to the two optic nerves. So these two big bundles here, this one and this one, will actually run out to the eyes of the animal. And they're really just a big bundle of those axon fibers. If you remember our brain cells, they have those big long tails. That's what these are, big, huge, thick bundles of many axons that are relaying information to the brain. Your olfactory bulbs are here. Those would actually bring in scent information. So what you're smelling is being relayed through the olfactory bulbs. Our first task here to actually get a really good look at this brain is to remove that dura mater so we can get right down to the tissue of the brain itself. So what I'm going to do is make some cuts through the midline and remove both sides of the dura mater so we can get down and see what we call the cerebral cortex. So with very small snips of my scissor, I'm going to enter here at the olfactory bulb, and I'm going to go around using that blunt end of my scissors to enter so that I'm not damaging the brain tissue underneath. Like I said before, this tissue is a lot harder and firmer than it would be if this was a fresh brain that had not been preserved. But a fresh brain would fall apart in our hands. So we can take that dura mater off of here and expose the cerebral cortex. So this front part of your brain, as we'll see once we get our dura mater fully removed, and we'll come down the midline with this as well. So now we've got our brain fully exposed. So looking at it now, there's a few things that we can see. The main part out front is your cerebral cortex. You can see it's organized in two sides. You've got your right side and your left side. And this is really the part of your brain that does a lot of the typical things you think of your brain doing. So your thinking, your problem solving, memory storage, language, motor control happens in here. So motor control is if you're telling yourself to move, you're going to be using these parts of the brain. Sense processing, a lot of that happens up here. But all of your real higher order thought, the things that make humans special, really happens in this outer area of the front part of your brain. We tend to have a bit of a bigger lump in our cerebral cortex here than the sheep's brain would. The sheep's brain's a little narrower here, but the same organization is the same, and the basic functions are the same. If we look towards the back of the brain, we notice what we call the hindbrain structures. And the hindbrain here is really this big lump called your cerebellum. And your cerebellum is the part of the brain that helps coordinate movement. So we said you might decide to move over here, but it's the cerebellum of your brain that figures out, okay, how do we go about that? How do you walk? You might decide to walk down the street, but it's your cerebellum that's going to coordinate all of those complicated muscle movements to do that. Even sitting in a chair is very difficult, and it's your cerebellum that has to control all those little things that are going to keep you balanced and sitting, and remember how to do all those things. So some of that muscle memory and that motion memory is stored in your cerebellum as you move around. So when things go wrong in a brain, whether it's a stroke or chemical imbalances or injuries, we need to figure out where in the brain these things are happening. You'll notice a lot of blood flow through the brain. That's these dark lines is blood flow. When we talk about having a stroke, that could be a lack of blood flow, so a blockage in one of these vessels or a smaller vessel inside that might block blood flow to one area of the brain. And whatever area of the brain is affected, we're going to see those things in that patient. So it could be language, movement, memory, things like that. When we're talking about Alzheimer's disease, it's that hippocampus, which is buried deep down inside. So to get down to that hippocampus, we're going to need to get in a little closer. If you look at this diagram, you can see it outlined in red, and you'll notice it's kind of located behind where the ear would be, off to the side in that brain. So the first thing we're going to need to do is make what we call a sagittal cut. We're going to cut this brain all the way through so we can separate the right side from the left side. It won't fully expose the hippocampus, but we'll be able to see some of the structures of the midbrain. So I'm going to hold this out of the way of the scalpel, and what I'm going to do is make some cutting motions through the entire brain. The main rule with the scalpel is that we draw, not saw. I don't want to be cutting into myself, so I'm making nice drawing motions carefully through this brain so we can make a nice clean cut. And basically what we want it to do is we want it to open just like a book so we can see those structures of the midbrain. And one last little bit here. Okay, so now we've got our brain in two, and we can see a lot of the structures that make up our cerebral cortex. That's this outer layer here that we were talking about before, all those things we think of our brain really doing actively. We see our cerebellum that again controls coordinating movement. We can also see a few other structures in here, such as your corpus callosum, which will link the two sides of the brain. There's our pituitary gland down here, and our brainstem and spinal cord. We can't actually see the hippocampus yet. Remember, it's buried on either side of the brain. So to do this, we're going to have to get a little closer. I'm going to put this piece aside for a second, and I'm going to use this side as well. You might notice there's two different kind of colors that you see when you look in the brain. You see the kind of this whitish color, and then you see a bit of a browner, darker color as well. There's a reason for this. This is what we call our gray matter and our white matter. The white matter that makes up the spinal cord, a lot of these branches you see in here, are axons of nerve cells. So you can picture those long blue axons on our nerve cell model. They're all bundled together like this, whereas the bodies and dendrites and a lot of those would be up where the gray matter is. I'll take this brain and just make a slice. Here. And we can see that gray matter, white matter is preserved throughout the brain in that structure. Okay? And it's all about the axons of the brain are coated in a white protein called myelin that helps conduct electricity. And we can see that in these brains as well. Okay? So let's use this half over here to see if we can find that hippocampus. The way I like to do this one is we're going to come down from the top of the brain in very thin slices. The hippocampus in you and me, like we said, is about the size of your pinky finger. In the sheep, it's obviously going to be a little bit smaller. The word hippocampus actually comes from the Greek meaning seahorse. So a lot of people thought it kind of looked like a little curled seahorse. I think it looks more like a banana, but as we make our cut, what we're going to look for is we're going to keep looking until we can find the head of that seahorse. So I'm going to try and make some very thin cuts from the top down here. And what we'll do is we'll just stop every time and we'll look and see if that little head of the seahorse is popping up. Right now all we see is that nice uniform gray matter, white matter pattern. Okay? So I don't see the head of the seahorse popping up yet. Let's go take off one more slice. Again, we just really see that gray matter, white matter pattern of the cerebral cortex. Okay, so let's take another slice. Here's where we want to go nice and thin, because we don't want to take the top of the seahorse's head off. We want to be able to get that hippocampus out exactly. Okay, still nice and uniform. Still cerebral cortex we're looking at. Okay. Go a little deeper. I still don't see that little seahorse head popping up. It'll be about pea size when we get there, about the size of a pea. Still fairly uniform, but you do see a little change just in here. That's telling me I'm probably getting close. We're probably through the cerebral cortex now. So I'm going to make sure that this next cut is about as thin as I can make it as I move through that area, because I have a feeling we're close. There. So if we look closely, can you see how something has changed? Now we can really see that's just the top of the seahorse's head. That's the very top of the hippocampus poking out into the surrounding cerebral cortex. So that's what we want to get out of here. Obviously, there's so many different parts of the brain, but when we talk Alzheimer's, it's really starting here. So let's see if we can pull that out. I'm just going to clear away some of the surrounding tissue. And what we're looking for is that seahorse or banana shape to come out really clear for us as we peel this brain apart. Okay, as we bend back the surrounding cortex, keep your eye on that seahorse's head right there. I'm going to peel that. Okay. We can see that structure starting to reveal itself as we peel away some of that cortex. Okay, there's our seahorse's head, and we can see it wrapping around. And as we peel back more of the surrounding brain, I'm being a little rougher here than I need to be, but we want to make sure we get that out intact. There's the head, there's that nice curve, and it's its own little structure within the brain. And it's defined well enough that we should be able to pluck it right out of here. And there we go. So, I don't know, you can decide for yourself whether you think this looks more like a banana or a seahorse. But that's our hippocampus. Okay, there's another one on the other side of the brain. You've got two as well, one on each side. And you look at this and you kind of think, this little weird white lump of stuff. Okay, this is actually every memory that you have, every memory that this sheep has been formed by running through this hippocampus. Remember, it's the one that helps you figure out where you are in space. It's the one that helps you decide how to take a short-term memory and keep it as a long-term memory. And when we talk Alzheimer's, we're really talking about the hippocampus. Neuroscientists here at the Research Center and around the world are trying to understand how this functions and how to stop that progression of nerve damage, those plaques and those tangles that seem to happen here. The brain really is one of the most amazing, mysterious, and important organs in your body. There are many things you can do to keep it healthy. Right now, there's no guaranteed way to prevent Alzheimer's and other neurodegenerative disorders, but we do know that there are many things you can do to keep your brain healthy. Eating healthy foods, exercising, reducing stress are important for your brain, as are specific things like wearing a helmet when doing sports or biking. Keep your brain active. Challenge it by reading, playing, listening to music, doing puzzles, or meditating. You can do things right now at your age that can benefit your brain for the rest of your life. Thanks for tuning in to Healthy Curiosity. Stay curious.
Make sure to click like and subscribe for more videos and follow us on Instagram and Twitter. To support the Youth Biolab's ongoing programming and expansion plans, please contact St. Boniface Hospital Foundation at stbhf.ca.